people got this point, and 11 people did not get this point right here. And if I go back and just kind of think about what I was reading, a lot of it was like not even talking about structural features of the plasma membrane. It was like, well, there's an axon and it's the postsynaptic one and stuff like that. No, we're just looking for, what creates and establishes the membrane potential as a structural feature of the membrane. So, questions? Yeah, there we go. All right, the second one is the exact same graph. And if I'm right, this one was even worse. 13 people didn't get any points. One person got 1 point and one person got 2 points. This is a 2-point question, okay? And what this is asking you to do is connect what's happening here on the top graph to what's happening here on the bottom graph, okay? And it just did not go well for most people. But actually, it should be, because the graphs are lined up, and they do that on purpose. They were like this. It can be easier than you kind of think, right? So this graph is going up. Membrane potential is going up, and that happens as sodium ions are entering the cell, or as the membrane becomes permeable. So that means sodium is causing depolarization on the uphill graph right there. And then the movement of potassium ions out causes repolarization. Those are how you connect the relative membrane permeability data in figure 1 with the changes in membrane potential in figure 2. And that's exactly what it says right here. It's about figuring out how those graphs line up. And Brittany, I think you did pretty well on that answer. So if I'm right, I can't remember exactly. But that's, that's.
Yes.
Yeah, excellent. Good, because that's a hard question to answer. Although I don't think it has to be a 13 right there, because it's really just let's connect these two things right there. So, all right. Today's goal, let's see, we are right here. So we're going to go over the test. I'm going to show you guys the spinach disc photosynthesis thing here in a second, and also some pigment chromatography, although I have to mix my chemicals here. That shouldn't take me very long. We're going to finish up photosynthesis notes, and we will have time to finish up the lab. Today, SmartBook Chapter 7 is due, and on Thursday, the virtual lab on photosynthesis is going to be due, that we'll have some time to finish up today. We will take a quiz on Thursday before the end of the semester. I'm just going to push that grade to the next semester. It'll be the first grade of the next semester. And we'll also look at the mystery of the 7 deaths and take some notes on cell respiration. The test for this one is actually coming up really rapidly. It looks like it's going to be on February the 5th. I really wish we had one more day, but that puts it at 3 weeks. And so we have last week, this week, and then next week. It's just that we're missing that third day because you guys have Monday off. It would be Monday, Wednesday, Friday. Instead, it's just Wednesday and Friday, I think is how it's going to be. So we're going to power through. We're going to try to get through all that because we have units on DNA and molecular biology and genetics coming up. And the AP test is going to happen, so I don't like changing my calendar at all. We may have to cut out respiration with CO2 meters and insects. I might have cut that out anyway, depending on how many people were here. But we'll see. I don't know. On Thursday, I'll know what we're going to be doing next week. So, all right. Today we're going to try to figure out what about plants that are not green is kind of our big question. Like, what's up with that? How does that work? So I'm going to show you all 2 things here. What was that, like Friday? Last time I saw y'all, we made spinach discs sink. And then by removing the air, then we made spinach discs float because we made them do photosynthesis. They floated by the end of class. It really didn't even take the whole class. I put them in here, and they continued to do cell respiration, so they used up their oxygen. And they sank. The question is, did they sink because they're dying and dead, or did they sink because they used up all of their the oxygen gas was making them float? And so let's put them in the light for the day and see if they refloat today. I actually have no idea what's going to happen. We'll find out. The next thing I'm going to do, I need to go get a beaker. got this and this. Okay. I bought this at Home Depot. I don't think I'm actually supposed to have it up here, but I have it anyway. It's petroleum ether. Really flammable stuff, smells like gasoline. And we're going to take a look, and I'm going to set this up while we're talking about photosynthesis. This is chromatography. Okay, Brittany, do y'all talk about chromatography in AP Chem?
We did a little bit.
So it's just a method of separating things based on any physical characteristic. There's tons of types of chromatography. This is called paper chromatography because we're going to use paper to separate out pigments. And you can see in this picture, on the left-hand side, we've got the setup where you add some plant pigments to this piece of paper, and then you dip the paper and suspend it over and dip the bottom of the paper into this solvent. The solvent that I'm using is very, very, very non-polar, which is actually going to matter. But let me try to measure this out real quick. Okay, so we want 18. All right. Again, I don't know if I'm supposed to have this one either. This is acetone. Okay, something that's another non-polar, or this is polar solvent. So this chromatography solution that I'm making is 9 parts petroleum ether, 1 part acetone. I'll explain what that means here in a second. It always gives me a headache after a day of smelling this, but it's just one class and then I can get rid of it. All right, so I'm taking my chromatography solvent here, and I'm pouring it into my beaker just as a nice place to hold it. And now I need some spinach. I continue to use my spinach here. I'm starting to smell that too. I'll have to get rid of it. And I need something narrow, but not too narrow. Usually I would use a coin, but a coin is actually not great. It's a little too narrow. I need to get the pigment out of the leaves and onto, I'm going to use the edge of this, and onto this paper. The idea behind this is, like, there are alternate pigments of photosynthesis. They're not all chlorophyll. The other ones exist for a reason that we're going to talk about once we get to the notes. But if we want to see them, we have to separate them based on their chemical characteristics. And that is basically their polarity. Because this paper is very, very polar. It's made of cellulose. The solvent is very non-polar. It's made of essentially like gasoline extract kind of stuff. And so what we're going to do is see which ones will dissolve better in the solvent and which ones will stick better to the paper. As that solvent soaks up the paper, the chemicals, if they dissolve well in the solvent, will travel a long way. If they don't dissolve well, and if they're highly polar and stick to the paper better, they will not travel as far. And so as that clear gasoline-smelling solvent moves up, the pigments move at different rates depending on their polarity. And we should be able, if I get enough pigment on this piece of paper, which I'll show you in a second, we should be able to see them in an hour or so. See all that chlorophyll, all that green? See all that green? Now all I'm going to do is take this and let it sit. Sometimes I have to bend it over a pencil because of the... Okay. The solvent is soaking up to the green right there. And in an hour or so, we should be able to see those pigments as they separate, and then I'll show you all those. But we're just going to let it sit right now. So, all right. And we've got spinach discs sitting. We'll see if they start to rise as they continue to do photosynthesis. That's the last time we're going to do that. and we've got pigments being separated based on their chemical characteristics. And that means we need to talk about photosynthesis. So let's talk about photosynthesis. Right here and here. Okay, we finished up and we had talked about kind of the introduction to photosynthesis, which is weird, not the introduction, I guess, the main big parts. We talked about the light reaction and we talked about the Calvin cycle right here. Okay, so now I want to kind of complicate photosynthesis, right? I want to see other things about it. And so we're going to look at reconciling the absorbance spectrum of chlorophyll and the action spectrum of photosynthesis. We're going to look at alternate mechanisms of photosynthesis, and we're going to look at the problem of photorespiration and then how to solve the problem of photorespiration. Basically, we're going to take regular photosynthesis and see what's going on with making it a little bit different. Okay, you have to recognize this graph. Okay, no matter what, like this is one of those things that I think tends to show up on tests. At the bottom, do you guys know what those represent? Any ideas?
Red, orange, yellow, green, blue.
So I flipped it just for wavelength purposes because, like, we say Roy G. Biv or red, orange, yellow, green, right? But technically, and if I did nanometers, you might see this in nanometers, but you would see like really small wavelengths.
Yeah.
And red has a much longer wavelength, right? And so I flipped it because it's the typical way that it would be seen on one of these. The y-axis can change, okay? Let's say for the first y-axis, we're going to do the percent absorbed. So that goes between 100 and 0. And we're going to draw a graph that represents, I'll even use green for this, it represents chlorophyll. And this is going to be chlorophyll's absorbance spectrum, right? It's going to be a graph showing how well chlorophyll absorbs different colors and reflects color, because if the line is down low and absorbance is 0, then it's reflecting that. So for chlorophyll, where is this line going to be the lowest as a green pigment? If we draw a wavy line over this that goes up and down, and it represents the absorbance spectrum of chlorophyll and how well chlorophyll absorbs and reflects different colors, where is the line going to be the lowest?
At green?
Yeah, because it's reflected, because it's what you see. And so this is a really rough approximation of chlorophyll's absorbance spectrum. It's actually a lot, it's much more specific than that. But that at least is an approximation. It absorbs red really well, and it absorbs blue, indigo, and violet okay. And so it doesn't absorb green, which is why it's green. And this, by the way, over here, that's chlorophyll. The reason it absorbs and reflects those colors has to do with the wavelengths of light and how they resonate and change the bond lengths, I guess, in the complicated chlorophyll molecule with all those loop structures. Most colored pigments have what are called aromatic ring structures like that, where they have like hydrocarbon rings. It's just like a common thing to see, but that shape absorbs red and blue pretty well. But that does not match up.
I have a question.
Yeah, go ahead.
If you were to grow plants without visible light, so you would do either like a specific color of light or a totally different light entirely, would it change the color of the plants itself?
Okay, so let me address your question after we look at this next one, okay? So it's a good question. Like, you're one step ahead of it. I want to put one more line up here. Okay, so this is not a single pigment. This is what we call the action spectrum of photosynthesis. It illustrates how the rate of photosynthesis can change depending on what color of light you use, which is usually white light, which is a combination of all of these, but we can get light bulbs that only shine like one of these colors, like you're asking about, right? So let's say, for instance, that we choose to shine whatever this wavelength is right here, right? So we're going to use that color light and only that color light to shine on the plant. Is chlorophyll absorbing that color of light? Is the green line high at that point? Yeah, it is. So chlorophyll is absorbing that light, and photosynthesis is happening, right? Because the action spectrum is like high up on the graph right there. So that makes sense, right? Now, what color would it grow? Not as well as it grew in like white light, but it would grow. And what color would it be? If you shined this one color of light on it and it absorbed all of that, I would assume that it would be gray, you know? And if we grow plants in the absence of green light, which you can do just fine, they look great because they're not reflecting that light back at you. They're absorbing all the other colors and they would look great. Would they grow? Yes, but they would grow slower because they're doing photosynthesis slower. Anybody notice the weird thing that's going on here in the middle? It doesn't make any sense. If I grow a plant in just green light, is chlorophyll absorbing that light?
No.
Yes.
See it's not. It's the green line is, oh, y'all can't see this. The green line is really low right there. So if I grow a plant in just green light, chlorophyll is not absorbing that because the line is really low. It's down here at close to 0% absorbance. Is photosynthesis happening if I just use green light?
Yes.
Yes, but not at such a high rate, but it is happening, right? But that doesn't make any sense because we already said that to start photosynthesis in the light reactions, you have to have like the absorbance of light to split water to get the electrons out. So how can we, what we call reconcile, right? How can we explain the difference if chlorophyll is not absorbing the light, but photosynthesis is still happening?
There's another pigment that's not.
Which is
not
what we just set up, right? What other colors of plants have you seen at certain times of the year? What other color do leaves turn?
Oh, red and orange.
Red and orange. So the orange one is going to have its lowest, its trough, I guess, right here, and then it peaks right there. And that, by the way, is known as the carotenoids, or you might know it as beta carotene, or the vitamin that we need to get from things like carrots, and it's orange in color. The red ones are, I think they are, there's like anthocyanin, which is kind of, I want to say kind of red, but if it was red, it would look like this. And there's yellow ones that are called xanthophylls that look like this. And so there are other colors of pigments that help with this. And the overall goal is like this black line right here. And this is an exact worded question from an AP question. If there were no other accessory pigments, alternate pigments, or secondary pigments, what would the action spectrum of photosynthesis look like if the only photosynthesis pigment was chlorophyll?
It would dip down at the green.
Good. If the only pigment was green chlorophyll, right here, if that's the only one, and if there was none of this, none of this, and none of this, how would the black line, which is showing you the rate of photosynthesis of different colors, look? And it would exactly match this. But it doesn't, right? Because of those other pigments. And that means accessory pigments are increasing the rate of photosynthesis and allowing plants to make more sugars and grow faster, and it helps plants a lot. We only get to see these things in the fall because it turns out chlorophyll, this molecule over here, it's really, really water sensitive. It starts to break down in the absence of water, and when plants go dormant, they shut off the flow of water to the leaves, and it starts to break down, and then you can see these guys, at least temporarily. They're usually there, but just hidden by the total amount of chlorophyll, which I'm trying to visualize for you guys with this, but it takes a little bit of time. So I'm going to keep this thing just hanging out right here.
Okay, I have another question.
Yeah. Okay, Brittany, let me answer this one first.
Okay.
What about like colored plants? So what do you mean by
Okay, cool.
A red rose.
Okay, that's a flower. That has nothing to do with photosynthesis, right? Red roses have green leaves. But that doesn't mean these don't exist, because think of like purple cabbage, right? Purple cabbage is the colored leaves, that plant, for one of a couple of reasons, either because it was specifically bred to be that way. It may not be like a natural like thing. It could have been a mutant that we found and we bred to become more purple and more purple and more purple. That's artificial selection. We talked about that with evolution, right? It still has all of these, but what we did through artificial selection and breeding, we just upped the red pigments and reduced the other ones. It probably doesn't grow as well. Like if you were to remove people from this and stop people from breeding red cabbage and red cabbage and red cabbage and things like that, it would probably not grow as well as the green one because chlorophyll is the one that is usually in doing this. So it's a good question, but you can't think about roses because the flower petals have nothing to do with it. Those are for attracting bees. Brittany, what was your question?
So if you grow it in one color light that's not green, it's
Just
going
like blue,
to
right?
be like gray or black or something. But what if you then take that plant and then put it in like sunlight? Would it then turn green or would it stay gray or black or?
It would. Okay, the reason, like let's say we grew a plant in just blue light, right? And we hang out in this room with a blue light bulb and we watch the plant as it slowly grows, right? Okay, is that plant still making chlorophyll?
Yes.
Yes, it is. So that plant still has chlorophyll.
Wait.
We don't see chlorophyll because there's no green light being reflected, right? And so it looks green to us because there is nothing for chlorophyll to reflect. It's just absorbing that blue light. Does that make sense? And then we take that plant outside, that chlorophyll is still there. So as soon as we take it into natural light, it will then have light to reflect. And so, I mean, unless there's some sort of controlled gene expression rate of chlorophyll production, I don't think there is. I think chlorophyll production is probably one of those genetically controlled things that is a set standard that is not environmentally controlled, if that makes sense. We'll talk about environmental control of gene expression later on.
I have a question too.
Sure.
Okay, so if plants have specific like things that absorb light and don't, but that helps for photosynthesis, do animals and stuff that don't need photosynthesis, they still have specific colors that show?
Yeah, and so like if we wanted to, I've never done this before, but let's see what happens with this Google question. Human skin pigment. The absorbance spectrum of melanin. Okay, so it looks like this spectrum includes things that are infrared and maybe even not UV because it doesn't absorb UV very well, which is why that causes sunburn. And so we make our pigments too, right? Our pigment happens to be kind of, I don't know, tan to brown in color. And different skin colors occur because of different, not different pigments, but different amounts of melanin. And so that would be the absorbance spectrum of melanin on that sloping line. And you can see that it kind of absorbs all colors, but with a little dip on the right side towards the longer wavelength reds and oranges, and greater absorptions of blues and purples. And so that's our pigment. That's why humans look like they look. And then the difference in like darkness is just an amount of, a factor of like how much.
So
But
non-living things still have like those extra pigments and stuff?
what do you mean, like non-living things?
Like rocks.
Every single thing that has a color, like the word pigment just means something that has a color, right? And so like if we were to go like red paint, right? Like red paint is red because there is a molecule in there that absorbs orange, yellow, green, blue, and purple and reflects red. Whatever that molecule is interacts with light and wavelengths of light to reflect that. And so, yeah, the word pigment isn't something special. It's just something that interacts with wavelengths of light to absorb some and reflect some. So yeah, living things and non-living things. What's special about chlorophyll is that it does a really good job of using that energy that it absorbs to split water so they can get electrons out. And that's relatively unique.
Yeah. Okay. Thank you.
Yeah. I'd never looked at that one before. That was an interesting one. I wonder if, I don't know the exact, but if we look at this, this is the melanin, and if you see how it drops down right here, I wonder what wavelength of light is most commonly associated with skin cancer, because the reason it causes skin cancer is we don't absorb it, and so I'm guessing something less than 200 nanometers, because you can see how in this graph at least it drops off really fast right there, which is why we have to be careful. And then sunscreen is going to absorb somewhere down here in the UV region. So I don't know, I've never really thought of it like that, but there it is. All right, so we need to think of another, like there's a problem with photosynthesis here, okay? And the problem is, and I'm gonna just do a simple little sketch right here. The Calvin cycle, it is going to need more ATP than NADPH. Okay, we looked at that when we went and did this right here. You can see that it needs 6 ATP, 12 ATP, so 18 ATP and 12 NADPH in order to make 1 round of this, or technically this would be like 6 rounds to make a sugar. Yeah, it's 6 rounds to make a sugar. Okay. The problem is the light reactions. These things make relatively equal amounts of NADPH and ATP. And that means that if the light reaction just continues as we talked about it, that it's going to produce either not enough ATP or an excess of NADPH. And those are both bad things, because like that's just not how photosynthesis in a chloroplast is supposed to work. And it's not like they can pump that ATP out for use in the rest of the cell. It just doesn't work like that. So we've got a problem where we have the light reactions produce equal amounts. Okay, let's fix that problem. I'm going to go back to this picture so I don't have to redraw it. Okay, this is how we did the light reactions, and we'll do this in yellow. We said the electron flow goes from here, water being split, here, and then here, and then it goes to NADPH. Okay, the origin of the electrons is right there. The final destination, at least right now, is right there. And that is called the Z scheme, okay? Because the electrons, if you turn your head like I said to the side, look kind of like a Z. That's, like, we need to skip one of those parts, right? We need to make sure that we can make some ATP, but not make some NADPH. And so there is this thing, and I'm going to switch to a different color that'll show up on this, kind of pink. This one right here, that is called non-cyclic electron flow, because the electrons go in a straight line. But there's a whole other way you can do this, right? You can have what's called cyclic electron flow. Okay, and this does not require water for it to happen, at least like at the beginning. Like you need 1 molecule of water and then you can just keep doing this. If I overlay on purple on top of this, let's say that you split water 1 time, right? You take those electrons and they're being passed from protein to protein, reaction to reaction to reaction, and they get down here to photosystem 1. Photosystem 1, when light hits them, can boost them up, and then instead of being passed to NADP plus reductase, where they end up at NADPH, they can be sent back to the electron transport chain molecules, and then you can just keep doing this kind of triangle right here, where we go this way, and this way. And plants can turn this on or off whenever they want. And every time it makes a loop, we get more hydrogen ions pumped. What do the hydrogen ions allow us to produce?
Electrons?
Nope. Remember this thing down here?
Water.
ATP synthase is the protein that makes ATP, and it is powered by a hydrogen ion concentration gradient. So by doing this cyclic electron flow, every single time it does that, it's pumping hydrogen ions back into the chloroplast, or back into the thylakoid membrane in the chloroplast. They can flow through, and then we can continue to make ATP, but we never actually pass the electrons on to NADPH. So plants can do one or the other, and it doesn't make any difference which one they choose to do. Okay, they do, when they need more, when photosynthesis needs more ATP, they turn off non-cyclic electron flow, they turn off the Z scheme, and they go over here to cyclic electron flow. The point of this is just to say, like, plants need things, and then they can change this reaction to create and make what they need. And if they need more ATP, then they need to make more of this hydrogen ion concentration gradient. I really want y'all remembering hydrogen ion concentration gradients and ATP synthase. That's about to show up on Thursday when we start talking about cell respiration. It's a huge part of that. So, all right, that's just one wrinkle right there. Our second wrinkle has to do with this. Right here. You see that right there? Carbon dioxide. You see that right there. How many carbons do you see in ribulose 1,5-bisphosphate?
5?
5. And I see 1 carbon in carbon dioxide. So that means over here, we would have, technically we get this carboxylase intermediate with 1, 2, 3, 4, 5, 6. So we get 6 carbons, right? That's good, because 6 carbons means we can start making sugars, because you have fixed carbons. And this enzyme that we talked about, known as Rubisco, is really good at doing that. There is a major problem with this enzyme. That major problem with this enzyme is it can do this entire reaction with oxygen 2. Okay? All of this will work with oxygen 2. But if ribulose 1,5-bisphosphate has 5 carbons and oxygen has 0 carbons, is there any fixation of carbon going on? Can we ever make a sugar if this happens with O2 instead of carbon dioxide? Is there any net gain of carbons? There's none. So this is an absolute waste. It happens all the time in plants, and it's called photorespiration. That means no carbon fixed. That means no sugars made. I need a pen so I can start making this a little bit neater. All right, so photorespiration, really, really, really bad thing. And here's when it happens. Okay, so let's take a leaf. That's a really bad leaf. And we're looking at the bottom of the leaf. Okay, there are all these little veins, branching veins. It's a dicot. Remember dicots and monocots, all that? And there's all these little pores. Now, this is a lab that we do later on, but I like to introduce it now because it explains what's going on with that lab later on. That pore is called a stomata. And it can look like either this, and somebody usually says a Pokemon ball, I guess, or it can look like this. It can be open or it can be closed. These things are really important. One thing is going in and 2 things are coming out. What can go in to that plants need?
CO2.
The what?
CO2.
CO2. Carbon dioxide is going to go into the leaves for photosynthesis. What 2 things are going to come out of that? This is into the air, right? Because this is leaves in the air.
Oxygen?
Oxygen. Anybody remember the other one that we talked about way back at the beginning of biochemistry and characteristics like adhesion and cohesion? What else?
Water vapor?
Water. Good. That's really important for plants because when a water molecule right there evaporates out, it pulls this one, which pulls this one, which pulls this one, and 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 this one. All the way down to the roots, water is being pulled up in a process called transpiration. Okay, you with me so far? That's how plants get water up. They let some water evaporate out, and that brings more water up, and they can bring water up to the leaves. There's a problem here. When would plants want to close their stomata so that this doesn't happen?
Whenever they are using up oxygen?
The oxygen and carbon dioxide don't matter here. It's water.
Oh.
When would plants want to close their stomata related to water loss? Or I should say, what climates or what conditions in the environment? Hot, dry conditions. In hot, dry conditions, plants try to conserve water, so they close the stomata, right? They close their stomata, water can't leave, they keep what water they have, and that's good. But all of a sudden, that arrow stops and that arrow stops. And last week I said they can't turn off photosynthesis, right? So if they keep photosynthesis going during the day in hot, dry climates, what happens to the amount of carbon dioxide in the leaf cells as photosynthesis continues and they can't bring in any more? And what happens to the amount of oxygen in the leaf cells as they do photosynthesis and they can't get rid of it? To conserve water, they close stomata, CO2 levels go down, oxygen levels go up, and this happens. Oxygen levels right here get so high that the carbon cycle, the Calvin cycle, same thing, it happens with oxygen instead of carbon dioxide, making no sugar, and all of this NADPH is wasted, and all of this ATP is wasted, and all of this ATP is wasted, and no sugar is produced because it got too hot and dry. And they can't turn this process off. Like, this will kill a plant. And this explains why, okay? Because they try to save their water. But that means plants that live in hot, dry climates, like sugarcane that grows at like equatorial areas, like pineapple, I'm just naming a few that I happen to know of. They had to evolve ways of fixing this. They need to evolve ways of fixing this problem right here of low CO2 levels and high oxygen levels causing the problem of photorespiration. Plants did it two separate ways. This is our last one. They have, by the way, most plants are called C3 plants, and they're called that because they make these 3 carbon compounds right there. But there are plants that are known as C4 or CAM plants, okay? These are plants that live in hot, dry climates, and I don't know which one is which, and it doesn't matter which one is which. The point is that plants evolved ways of getting around this. I think that this one is a little bit easier, okay? AM reminds me of what's going on here. This is a leaf cell, okay? In this leaf cell, we have the Calvin cycle that is taking CO2, putting it in, and we get sugar out, right? And we just said the problem was not enough CO2. So let's store carbon dioxide, but it's a gas. You can't do that. So for these guys, what they're going to do is at night, they store CO2 as an acid at night. So at night, they open up their stomata. They take in CO2, and then they turn it into this acid. It's called oxalic acid. It doesn't matter. but it's a 4-carbon acid. And then that can then be broken down into the CO2 during the day. So they have all this acid right here that makes them like store CO2. So they never drop with CO2 because they open up their stomata at night. And that's why it helps me to remember this. CAM plants means AM and PM, right? Like morning versus evening. And they do their photosynthesis during the day, but then they store carbon dioxide at night. C4 plants evolved a separate way of doing it. They separate out the Calvin cycle with CO2 going in and sugar coming out from the gas exchange portion. They have different cells. So up here, we get CO2 coming in. We make that same organic acid. That acid can be stored in those separate cells. And then this is like, if we were to physically look at this, like that's the outside of the leaf. And this is like the inside, like deep inside the leaf right there. And they have physically separated out. Both of them evolved separate ways of solving the same problem of storing CO2. One by physically separating the Calvin cycle from the gas exchange, and one by storing CO2 at night so that they can do it during the day. So this is temporal separation and this is spatial separation, both solving the problem of photorespiration. That's photosynthesis. Do you think we separated out some pigments? We did. Okay, there's this weird thing that happens here where we get, actually we get like 4 or 5 pigments. It helps to see it when it's dry. One of them shows up a lot better once it is dry. And I have no idea why that would happen. Like it's not visible when this is still wet, but it turns out this stuff is super easy to dry because it's like highly volatile. Probably really bad for us too. I'm gonna put this in the fume hood so we don't smell it. Before I show you guys while I let this dry, I want to show you the alternate pigment action spectrum.
Wait, so are plants in drier climates more acidic?
Okay, I've had that question before, and like, because that oxalic acid, it's a 4-carbon acid, so you get a 3-carbon acid, you fix CO2 to it. Anytime CO2 is in something, it makes it acidic, but like, is that why? I don't think that matches up, because it's not like acetic acid, like a vinegar or something like that. It's a good question, but it certainly is possible.
Pineapples are acidic.
Pineapples are acidic, but the bigger question with pineapple is that they have enzymes that, like when pineapple is like, you know, like when you taste all that, I don't know, when it hurts your mouth. Have you ever had that happen?
Yeah.
That is not the acid. That's the protein digesting enzymes that they have also. And so that's a completely different thing. But it is also acidic. And it could be that it's acidic, but are oranges, let me see. Hang on, I'm going to look up a question here while we're doing this.
Okay.
Are citrus plants C4 or CAM? But see, citric acid. Okay, I don't think this works because I just found this list on a, I don't know, ResearchGate website. C4 plants would include corn, sorghum, sugarcane, like grasses, because those are all grasses and grains. And then CAM plants are pineapple and prickly pear. And then C3 plants would be wheat and barley and pepper and fruit and rice and tomatoes. And there's plenty of fruit. I don't think it matches up. So it's not like the same like acid that makes things like tart and stuff like that. So no, it sounds like it would, but although I see, let me look this one up here as well, because our citrus plants see. Okay, so C3 are normal photosynthesis plants, right? That's wheat, barley, peppers, fruit, rice, and tomatoes. And those things, they're losing 40% of their photosynthesis like rate due to photorespiration. But because CAM plants and C4 plants have these like evolved processes, then they don't actually have any detectable photorespiration right there. So it just saves them a whole bunch of, I don't know, time and effort, which might be good because, they're in water-stricken areas, like desert areas, I guess. So I don't know. I thought that was interesting. All right. Before we look at this, let's take a look at the action spectrum, alternate pigment action spectrum. All right. Something like this. This shows all the different pigments that you can get. I'm not getting a great orange on this one. There's two chlorophylls. Here's an even better one. There's actually more than that. Chlorophyll A, chlorophyll B, carotenoids, phytochromes, phytochrome red, carotenoids. You can see all the different colors. They started out right there, and they moved up and they separated. There is an orange that's up here. It's just kind of faint. There's yellow, blue-green, olive-green, yellow, and then like a little bit of an orange at the very top. All right, let's see. I don't know how well this is going to work. Not well at all. There it is. Okay, so that's where I put the pigments. You can still see a little bit of a remnant, although it's more brown. And then this, can y'all see the yellow and the blue-green and the dark green?
Yeah.
Yeah. So they're there. They separated. There's even an orange that's like way up here. I'm trying to like hide the light from it. Hang on. Let me try this real quick. But this one works better. Camera. Stop presenting. I don't see my camera. I don't know. There it is. Oh, no, it's better. Yeah. Okay. So they're there. We separated out the pigments. We can see all the pigments from photosynthesis. All right. Let's turn this one off because that's weird. That means that with the rest of class, on whatever day it was, Friday, we had Photosynthesis 1, virtual lab. This is that John Darko guy that makes all these virtual labs. Wherever you are, we've got 30 minutes left. Let's try to finish this up. It is due at 8:30 on Thursday. Okay? Let's finish this.